Hello everyone, welcome to Positron Academy. Today the topic of discussion is on clinical radiobiology, in which we will study about the cell survival curves and the target theory. So the first question will be definitely, what is a cell survival curve? So a cell survival curve is a plot of surviving fraction against a dose, and this dose can be any radiation dose or can be any cytotoxic drug dose or any other cell killing agent. But as our topic is inclined towards radiobiological perspective, so you will assume that all doses that we are discussing over here are radiation dose. So in this graph you can see that as radiation dose is 0, the fraction of cells surviving are highest. But as we keep on increasing the radiation dose, the fraction of cell dying also keep on increasing, and after a defined radiation dose, the fraction of cell surviving becomes 0. So that's a graph or a curve which represents how with increasing radiation dose, the fraction of cell surviving changes. And to indicate the sensitivity of the cells, we define two terms which we call as ED50 or ED90. So what is ED50? So that is effective dose 50. That is the dose at which 50% of cell dies is called as effective dose 50 or effect dose 50. And ED90, the dose at which 90% of cell dies is called effect dose 90. There can be other representation also, like ED 50-60 or same goes for ED 90-60. What does it mean? Effective dose at which 50% population dies in 60 days, that is effective dose 50-60, or effective dose 90-60, the dose at which 90% cell dies in 60 days, that is effective dose 90-60. Now these values are being used for indicating the sensitivity of cells to radiation. Suppose we have two different groups of cell over here, and for one group of cell the ED50 values are 5 gray, and for other group of cell the ED50 values are 9 gray. So now we can say that this group of cell are more sensitive to radiation than this group, because at lower radiation dose this group of cell are dying more. So to define sensitivity of cells to radiation, we define two terms called as ED50 and ED90. So this graph is the same graph over here, but it is on a logarithmic scale, and this graph is on a linear scale. So this is on a logarithmic scale. So why this log scale is very important? So because it helps us to see and visualize the very low cell survivals. For example, we can now see the extremely low cell survivals over here in this cell survival curve, but that's not being visible over here. And why we need to see such low cell survivals? Because these low cell survival are very important in reduction of tumor size or even local tumor control. For example, after a non-curative radiation therapy, a tumor regrows. Why it regrows? Because post irradiation there are some stem cell left. Which stem cell? Those neoplastic stem cell, and those stem cell have the capacity to proliferate because they are colony forming cells. They are clonogenic in nature, so they regrow and make the tumor big again. That's why we should be familiar with low survival cells, because we don't want, after irradiation, we don't want any single stem cell to survive, because if survives it makes the entire treatment a failure. That's why we need to go for a logarithmic curve to look for the extremely low cell survivals, which are very important for reduction in tumor size or even local tumor control. Now the next question is that how radiation responds at the molecular level. So radiation basically kills cell by producing secondary charged particles and free radicals, and these secondary charged particles and free radicals in turn damage the DNA, and damage to the DNA is related to cell killing. For example, if you give 1 gray of low LET radiation, so these 1 gray of low LET radiation produces 1000 base damage, 1000 single strand damage, and 1000 or 20 to 40 double strand damage. And because of the absorption of this 1 gray of radiation by the cell, it leads to the production of these lesions. But not all the lesions are important. Some lesions are more important. Fine. For example, it has been observed that for radiation lethality, so number of residual and unrepaired double strand breakage post irradiation is important for radiation lethality. So if the number of residual and unrepaired double strand breakages are more post irradiation, then the probability that this therapy will cause more lethality will increase. So how much unrepaired double strand breakage the radiation has created, that is an important factor for predicting radiation lethality. So now the next question is, if we modify the cell killing. So this cell killing can be modified by different parameters, like changing the linear energy transfer of the radiation, or by changing oxygen level, or by changing thiol concentration, keeping fixed radiation dose. So if you go and change these parameters, so what are you changing? You are changing the amount of base damage you are making, the amount of single strand breakage, and the amount of double strand breakage. But what is more important? More important is the amount of double strand breakage. So whether you modify the cell killing or not, if you can increase the number of double strand breakage, then obviously you are going to make this therapy possible. So even if you don't go for it, so only the double strand breakage, it correlates with radiation lethality. So one residual double strand breakage or a single hit in a vital section of the DNA, it will be sufficient to produce a significant chromosomal aberration and thus to sterilize the cell. So we need to target on some specific regions, and that target to the specific region will lead to the lethality of the cell. And there comes the concept of the target theory. Now again asking the same question, that how radiation kills cell. So answer is again damage to specific region of the DNA that are more important to maintain their reproductive ability of the cells. And what are the specific regions of the DNA? So these regions are those regions which are the target for the radiation damage. For example, let us assume a hypothetical situation, assuming that's the DNA over here, and of this DNA, this part of the DNA is very critical for maintaining the reproductive ability of the cell. So if those secondary charged particles or the free radicals attacks this region and cause double strand breakage over here, then it will lead to the inactivation of the cell. But if these charged particles attack the over here, so it may not lead to inactivation, because that's not the target for inactivation. So radiation kills cells by targeting or damaging to some specific region which are the target for radiation damage. So based on the target theory, we have devised two different types of target theory. It can be a single target inactivation or a multi-target inactivation. So what basically happens in case of single target inactivation? So in this case, each cell has a single target, and if you inactivate that target, it leads to the killing of the cell. And this survival curve over here is a straight line on a semi-logarithmic plot of cell surviving fraction S against dose T. And to derive the equation of this curve, we have to use Poisson statistics. And how to define the probability of survival? So probability of survival means probability of zero hit. So as you mentioned before, that if this is a target, so hit over here will lead to cell killing, but if it does not hit, it does not lead to cell killing. So it means for survival there must be zero hit on the sensitive region, and that's equals to exponential of minus d by d naught. So what is d naught? D naught is the dose that gives an average of one hit per target, and the ratio d by d naught is the average number of hits per target. Now all the calculations for this target theory, or that is the hit probability, these are being governed by means of Bayesian statistics, because the probability that a given particle will hit the specified target is quite less, and that's why we need to rely on Bayesian statistics, where the probability of n events is given by P of n equals to e to the power minus x into x to the power n by factorial n, where x is the average number of events and n is the specific number of events. Now let us find the probability that the given cell will survive. So as I mentioned before, that for the cell to survive, it must have zero hit. So what's the probability of having zero hit? That is, when n equal to 0 and number of target site, that is average number of events, that is equal to 1. So when n equal to 0, when x equal to 1, then find the p probability from this formula, and you will find that's equals to 0.37 or 37%. So and d naught that we mentioned before is being called as the mean lethal dose, or the dose that delivers on average one lethal event per target. So that means if we deliver a dose of d naught gray, suppose, so if we give a dose of d naught gray, so what it does, it reduces survival from 1 to 0.37, that is it reduces by e to the power minus 1. So that is all about the single hit target model, and this model is valid for the inactivation of viruses and bacteria, but also valid for a mammalian cell to some extent in describing the very sensitive human cells. It can be a normal one, or can be a malignant one, or can be valid for radiation response at very low dose rates, or can be valid for radiation to high lead radiations. So in average, so in all, what this model is all about. So this describes a simple situation. If an given cell receives a dose of d naught, then there is high probability that the cell will die. And if the cell receives dose less than d naught, there are high probability that the cell will survive. That's all about the single target, single hit model. Now let us move towards the multi-target inactivation model. So these models are basically generalized for mammalian cells, and they have a shoulder on the curve, on the cell survival curve. So what is this shoulder? So these are the shoulder part. So it means that for cell to die, it needs a threshold radiation dose, and once that threshold radiation dose is being surpassed, then the events that lead to cell lethality keep on increasing, and the number of cells keep on dying over increase in radiation dose. So we can see that dq over here, we call that dq as the threshold dose, or we call it the quasi threshold dose, and this quasi threshold dose is related to this n by dq equals to d naught log n base e, where n is, we call as extrapolation number. So this dq depends on n. So if dq is large, n will be large. If n is small, dq will be small. Example, in this case, when dq is quite larger, then n is like this. When dq is smaller, then n will be somewhere like this. So that means that this dq depends on n, and n over here is extrapolation number. Fine. So for larger dq, n will be quite more larger. Fine. Now the question is why our mammalian cells in general have a large quasi-threshold region. So there are two different interpretations for this shoulder region, and these are that cell death results from accumulation of events, and which are incapable of killing the cell individually, but when these events keep on accumulating with time, and those lead to the lethality of the cell, and that's why cells start dying after a given dose. And second reason is that there when with increasing number of dose, the number of lesions which keep on increasing, and at when radiation dose is less, number of lesions are being repaired, but when dose keep on increasing, lesions also keep on increasing, and after some time the efficiency of the repair enzyme does not work that well to repair the lesions, and that time the cell keep on dying. So that's why it takes this amount of time for these to work out, and that's two models who describe why they have a shoulder region and why after this region the cell keep on dying, and the one being the target model and the other being the repair model. But now the question, now let us explain how this derivation is being done over here. So for probability of having 0 hits on a specific target, as we know, for probability of survival is equals to exponential of minus d by d naught, and probability that a specific target is being inactivated. For example, we know for multiple target model we have n different targets which need to be inactivated for the entire cell to die. So probability that one of the target will be activated, like this particle will hit this target, so that will be 1 minus exponential of minus d by d naught, because that this one exponential of d by d naught is for survival. So this is for probability that one target will be inactivated. So but we know in case of multi-target single hit model there are n putative targets. So for all targets to be inactivated, the probability becomes 1 minus exponential d by d naught times n, because there are n different targets. So and if all these n targets are being activated, that the cell will die. Now probability that cell will survive, then 1 minus probability of dying, so that's the probability of survival. Is it fine? So the next is the problem with targets. The problem is that it predicts a response that is very flat in low radiation dose. It means over this range there is no cell killing. Over this threshold or the shoulder region there is no cell killing. But based on clinical experimentation results, there are cell killing at low doses. So this model fails to include that one, the effect of radiation at low dose region, and that's why a different model is being proposed by adding a single target element which fixes the initial slope. So what we have done, we have included one more parameter d1, and this d1 fixes the initial slope to give into account the response at the very low dose region, but it still has a problem, and the problem is that the change in the survival over the range of 0 to dq occurs almost linearly. So change is almost linear. For example, if I go for a treatment with 20 gray, suppose total dose being 20 gray, so if I give this 20 gray in 2 gray per fraction or 1 gray per fraction, according to this model the response will be similar, but that's not the case. Whenever we fractionate dose, the response in low dose regions are not same. Response by 2 gray per fraction, response by 1 gray per fraction are not equivalent. So this problem has this issue, and that's why we introduce one more model which has a better vision than the previous one, which we call as the LQ model or the Linear Quadratic Model. So in this model we have two different parameters. One parameter is linear and the other is a quadratic parameter, and we can see over here the formula for cell survival is ln s equal to alpha d plus beta d square, and probability of survival is exponential of minus alpha d minus beta d square, or equals to exponential minus alpha d plus beta d square. And this model gives a better description of the radiation response in the low dose region, and that's the main implication of this LQ model, that it gives a better description of radiation response in the low dose region. and we can see the curve over here. It's continuously bending with no straight portion either at the low or high radiation doses, where we can see over here it has a straight portion over here, but these one don't have any straight portion, and it continuously keep on bending at either at low or high radiation dose. So how much bend it will be, how much bendiness this curve will have, it depends on a value which we call is the alpha by beta value. So what is this alpha beta value, we'll discuss in a bit. And before that also we can see that this graph does not have the D naught, because the survival curve continuously bends downwards with increasing dose, so it becomes never straight, and that's why we don't have a D naught. So now let us have a question that what we mean by the alpha by beta ratio. We can see over here in this area that the component of killing by means of linear component and the component of killing by means of this quadratic component becomes equal. So that is we call as the alpha beta, that dose of radiation at which the component due to this linear component and quadratic components become equal, we call as that is alpha beta ratio, or we call that is alpha beta value. So these alpha 1 is basically dominant at the low dose region, and it's because of a single electron track which intersects both the two adjacent chromosomes. So that's a single electron track which is intersecting both the chromosomes. And for the quadratic one, so it's a two track action, and it arises from two independent electron track and each intersecting one of the two adjacent chromosomes, and that's the probability that's equals to alpha d plus beta d square, and that's the curve over here, which is the LQ curve. So this curve is for high LT radiation and this one is for low LT radiations. Again, moving to alpha beta ratio as we have defined, so it's the dose at which killing by linear component and the quadratic components become equal, that is linear component dose is alpha d, and for quadratic components is beta d squared. So that dose at which two components become equal is the call is the alpha beta ratio. Fine. And this ratio, as I've told before, describes the bendiness of the curve. So how much the curve will bend, it depends on this alpha beta ratio. So this alpha beta ratio is thus the measure of how soon the survival curve begins to bend over significantly. And in the next slide we will see how this ratio influence the bending over here, and also that we can see that for early responding tissue and tumors they have a large alpha beta ratio, and for late responding tissue they have small alpha beta ratio. Example, for tumors they have alpha beta of 10, for prostate tumors they have alpha beta ratio of 3, and this graph will tell you about the influence of this alpha beta. So as I told before that these tumors and early responding tissues, they have more alpha beta ratio, so that the bendiness is quite they are lesser bending. So as the bendiness value, as for this one the value is lesser, so it bends more. So how much the curve will bend depends on the alpha beta value. So that's all for today, and in this lecture we have discussed about cell survival curves and the target model. Thank you for watching. Please like, share and subscribe to this channel and support us in making more videos. Thank you for watching.